Good morning students. In the last session we have studied introductory part of the urea cycle. Okay. Now in this particular session we will be studying in detail how exactly the urea is formed in the liver cells. Okay. There are 4 names of this cycle, which are those: ornithine cycle, because ornithine is the first non-protein amino acid which is formed in this particular cycle. Okay. And it is also known as urea cycle because main product of this cycle is urea, and this urea which is there, it is formed, it is highly, it is less toxic as compared to ammonia. So that is why our body converts ammonia into urea. Okay. Urea cycle because urea banta hai. Then ornithine arginine cycle because ornithine is the first amino acid which is starting this particular cycle, and arginine is the last amino acid of this cycle, because of which there is formation of the urea. Okay, got it? That is why the name given is ornithine arginine cycle. And lastly, Krebs-Hanselit cycle. Last time बोला था मैंने, why it is known as Krebs-Hanselit cycle? Because both the scientists have discovered this particular cycle in the liver cells. Okay, got it? So now let's study how exactly step by step there is formation of the urea, and which are the enzymes and how many ATP molecules are needed for formation of the urea. Okay, so firstly, reaction कहाँ पे start होगा? Mitochondria of the liver cells. So in the mitochondria of the liver cells, urea cycle will start. Okay. So carbon dioxide, 1 molecule of carbon dioxide and 2 molecules of ammonia will react with one another to form carbamyl phosphate, and this carbamyl phosphate is formed in presence of 2 ATP molecules. So 2 ATP molecules will get converted into 2 ADP molecules, and likewise there will be formation of carbamyl phosphate, and this reaction is catalyzed by enzyme carbamyl phosphate synthetase. Okay, carbamyl phosphate product का नाम है, और ये product बन रहा है because of reaction between 1 molecule of carbon dioxide and 2 molecules of ammonia. That's why synthetase word is there in the enzyme name. So very easy. Which is the enzyme which is needed for formation of carbamyl phosphate in urea cycle? Carbamyl phosphate synthetase. Okay, when there is reaction between carbon dioxide and 2 molecules of ammonia. And how many ATP molecules are needed for this step? 2 ATP molecules. Now this carbamyl phosphate which is formed, it will release the inorganic phosphate, and remaining part of the carbamyl phosphate will get reacted with the ornithine. So ornithine and carbamyl phosphate will react with one another in presence of ornithine transcarbamylase. Okay. So ornithine transcarbamylase will fasten this reaction, and likewise there will be formation of citrulline. Citrulline is also non-protein amino acid. Okay, proteins are made up of amino acids, but all amino acids will not form proteins. There are certain amino acids which do not form protein. Such type of amino acids will be known as non-protein amino acids. Okay, so ornithine non-protein amino acid hai, okay, which will react with carbamyl phosphate to form citrulline. Okay. Now this citrulline which is formed, in this citrulline amino group will get added up, and this amino group is given by another acidic amino acid, and that acidic amino acid is aspartic acid. Okay. So aspartic acid amino group उसका donate करता है citrulline को. Okay. And in citrulline also utilizes 1 ATP molecule and Mg2 positive ions, and likewise there will be formation of arginosuccinic acid, and for catalyzing this reaction arginosuccinic acid synthetase enzyme is needed. Okay. Likewise then afterwards whenever the arginosuccinic acid is formed, this arginosuccinic acid will then split up into arginine and fumaric acid. Okay. So, arginine fumaric acid is formed because of splitting of arginosuccinic acid in presence of enzyme arginosuccinase. Arginosuccinase. Okay. So, ये वाला एंजाइम ये फास्ट करता है स्प्लिटिंग किसका, अर्जिनोसक्सिनिक एसिड का, एंड बिकॉज ऑफ स्प्लिटिंग ऑफ अर्जिनोसक्सिनिक एसिड फ्यूमरिक एसिड इज फॉर्म. फ्यूमरिक एसिड क्यों बनता है, फ्यूमरिक एसिड इज फॉर्म टू ऐड द अमाइनो ग्रुप टू द एस्पार्टिक एसिड. अगर अमाइनो ग्रुप नहीं मिला तो अगेन एस्पार्टिक एसिड विल नॉट बी फॉर्म, एंड इफ अगेन एस्पार्टिक एसिड इज नॉट फॉर्म देयर विल बी नो फॉर्मेशन ऑफ citrulline. Got it? So, to regenerate the aspartic acid, fumaric acid will help. Okay. How fumaric acid will help? Fumaric acid will add the amino group to the aspartic acid. Got it? Now, this arginine, fumaric acid क्यों बना, all of you understood? To add the amino group to the aspartic acid. Now, this arginine which is formed from arginosuccinic acid will be used for formation of urea. So, arginine in presence of water, it splits up into urea and ornithine. किसमें split हो रहा है, urea and ornithine in presence of which enzyme, arginase enzyme. Got it? And likewise again there will be formation of ornithine. Okay. So, again the cycle will go on, and likewise many molecules of urea will be formed in the liver cells. So, we have studied here how urea is formed in the liver cells. So, remember students, urea formation will take place in the liver cells, and sometimes some quantity or else some amount of urea is formed in brain and kidney. But remember, elimination of urea is done by main excretory organ, that is pair of kidneys. So, kidneys सिर्फ eliminate करते हैं यूरिया, यूरिया formation का काम liver cells करता है majorly, and to some extent brain cells and we have studied that is kidney cells. Okay. किडनी में बहुत कम क्वांटिटी में यूरिया बनता है, बट वेरी लार्ज क्वांटिटी ऑफ यूरिया इज फॉर्म्ड इन द liver cells. Okay. So, let's quickly revise this again. Urea which is there, how it is formed? Due to reaction between carbon dioxide and ammonia. Carbon dioxide ammonia are both ways. These carbon dioxide ammonia will react with one another in presence of enzyme carbamyl phosphate synthetase, and likewise there will be formation of carbamyl phosphate, and in this process 2 ATP molecules will be utilized. Carbamyl phosphate will give inorganic phosphate, it will release inorganic phosphate, and rest all part of the carbamyl phosphate will react with ornithine in presence of ornithine transcarbamylase, and this ornithine transcarbamylase will fasten the formation of citrulline. Citrulline is also one of the non-protein amino acid. Citrulline will take the amino group from aspartic acid, and likewise it will also utilize 1 ATP molecule and Mg2 positive ions in presence of enzyme arginosuccinic acid synthetase, thus forming arginosuccinic acid. Okay. Now, further arginosuccinic acid splits up into fumaric acid and arginine. Fumaric acid is formed to regenerate aspartic acid. How it regenerates aspartic acid? By donating the amino group to aspartic acid. Okay. Aspartic acid is needed for formation of citrulline. So, that is why fumaric acid is formed. Okay. Now, other product which is formed due to splitting of arginosuccinic acid is arginine. Arginine, in presence of arginase enzyme and 1 molecule of water, there will be splitting of the arginine into urea and ornithine. Urea and ornithine. Okay. Likewise, urea is formed. So, arginine is the final amino acid which is leading to formation of urea, and ornithine is the first amino acid which is starting the urea cycle. That is why urea cycle is also known as ornithine-arginine cycle. Okay. So, in this particular cycle, total 3 ATP molecules are utilized. 2 ATP molecules in the first step and 1 ATP molecule between citrulline and arginosuccinic acid. Okay. But remember one thing, whatever 1 ATP molecule which is utilized by the citrulline to form arginosuccinic acid, here 2 inorganic phosphates will be utilized, thus converting 1 ATP molecule into AMP. How many AMP? 1 AMP. Got it? So, in this particular cycle, in this particular cycle, 2 molecules of the ammonia are utilized for formation of 1 molecule of urea. And how many amino acids are utilized? 3 amino acids are utilized for formation of 1 molecule of urea. Which are those? Ornithine, citrulline and arginine. Got it? So, this is all about urea cycle. So, here we have completed physiology of excretion first method, that is chemical part or chemical method. In chemical method we have studied deamination. In deamination we have studied 2, oxidative deamination and hydrolytic deamination, and now we have studied ornithine cycle. So, we have completed chemical part of physiology of excretion. Now, from next session onwards, we will be learning about the mechanical part of physiology of excretion. Okay? Thank you.